Hello, my name is Renata. I'm a master student and my research project is Pharmaceuticals as emergent contaminants are cytogenotoxic and capable of induction chromosomal variations. So, at first, pharmaceutical active compounds are chemicals used worldwide in prevention and treatment of disease. However, due to their overuse, large-scale production, and continuous emission, reports of these contaminants in aquatic environments have been consistent. They are considered ubiquitous in different water matrices and have been cause for concern among the scientific community. Pharmaceuticals constitute a significant class of aquatic contaminants and can seriously threaten the health of non-target organisms because they present an ecotoxicological potential. They are currently considered as emergent contaminants for which no strict legal regulations on the consumption and release of these pharmaceuticals into water bodies have been implemented on a global scale. A greater understanding of their effects on the environment are still required. And nowadays, the adverse effect on biodiversity are poorly understood. A previous study carried out in the region of Juiz de Fora, Minas Gerais, investigated the presence of pharmaceuticals in the Paraibuna River. This work showed that three different pharmaceuticals, metformin, valsartan, and atenolol, stood out with the highest detected concentrations. Because of that, these were the pharmaceuticals selected for this study. Also, this study also again showed the presence of this contaminant, highlighted the need for investigated regarding adverse effects. Then, the object of this study is evaluated mixtures of pharmaceuticals metformin, valsartan, and atenolol at environmentally relevant concentrations through the germination and hot growth assay in Lactuca sativa and their cytogenotoxicity in the models Daphnia magna, Drosophila melanogaster, Danurério, and Adel cepa, which are established models for genetic and ecotoxicological studies. To do that, and in order to expand the results obtained here for a global context, we are going to use a database from Germany called Pharmaceutical Environment, which has a record of the global occurrence of the pharmaceuticals in the environment, and to define the concentration tested in the assays, the entire range of concentrations identified in the database for the three investigated pharmaceuticals will be considered. The first test will be a phytotoxicity test on Lactuca sativa, through which will select the treatments that showed a significant difference in the hot growth to proceed with the other assays. And then, experiments will be conducted on allium cepa, such as cytogenetic analysis and flow cytometry. The endpoints evaluated will be parameters of cell cycle and cell death. Additionally, other techniques will be employed in different model organisms, such as comet assay, tunnel, and electrophoresis, which allow for the assessment of DNA fragmentation and other parameters of cell death. Therefore, through the evaluation of environmental risks caused by pharmaceutical contamination in aquatic environments, not only in Juiz de Fora but worldwide, we hope to enrich the scientific community, bringing new data on the ecotoxicological and cytogenotoxic potential of pharmaceuticals products at environment-relevant concentrations, and expand knowledge about the effects of this kind of contaminant, assisted in the development of pollution mitigation strategy. The results obtained here can contribute to the formulation of regulatory guidelines in Brazil and in other countries. Finally, I would like to thank all the partners and colleagues from the Laboratory of Genetics and Biotechnology from OFJF and other universities, and FAPEMIG for their financial support. I would also like to thank my advisor and co-advisor, Professor Lyderson and José Marcello. Thanks.